Hello, everyone. Welcome, everyone here in the audience and everyone online to this first Human Evolution and Archaeological Sciences seminar for the year. We're in the Human Evolution and Palaeolithic series today, and Philip Nigst and I have organized a great series of speakers. We're super excited. The first speaker today is Clement Zanolli, and he is a paleoanthropologist, a specialist in the study of dental structure of fossils and modern primates. He's a researcher at the CNRS, based at the University of Bordeaux in the Laboratoire Pasteur. And he did his PhD actually in the Museum of Natural History in Paris in France in 2011. And then following that, he did a range of postdocs in some very nice locations. He went to Trieste in Italy, and then he went to Pretoria in South Africa, back to Toulouse, and now he is, as I said, based in Bordeaux. He's a member of some really exciting paleoanthropological research teams in Africa. That's South Africa, Eritrea, Mozambique, as well as in Asia and Laos and the Philippines. And he's also an editor at the Journal of Human Evolution. His research projects cover a wide geochronological range, from the evolution of the Miocene hominoids to the origins of the genus Homo in Africa and to Asian hominin diversity in the Pleistocene. And that's the subject of his talk today. As you can see, hominin diversity in Asia during the Pleistocene. So very, very great welcome to you, Clement. We're looking forward to hearing your talk. We'll have some questions at the end. Anyone online can participate too. Merci, and have a great talk.
Thank you very much. So first, I would like to thank Tom and Philip to invite me to participate and to contribute to this HEAS seminars. I would like to also acknowledge that in the presentation I will show you, I was not the only one doing the analysis, but it's made in collaboration with many colleagues. And of course, there are also works that I didn't publish myself, but that will be used as a reference. So just for information also, I will focus mostly on the late Pleistocene, but I will also move here and there to the early and middle Pleistocene just to give you a broader view. Let's see if it's, yeah. So here you have a map with some of the sites that yielded middle to late Pleistocene human remains. It's not a comprehensive map, of course. It's just the few sites that I will mention today. And I will also try to go through all of these sites and try to let's give you a view, a taxonomic view of the human remains that were found. Please. So let's start with Homo erectus. So Homo erectus is a very long-lived species. We find it from the early to the late Pleistocene. And here I will just focus on a few sites just to give you an idea of the evolution of Homo erectus in Asia. So Homo erectus emerged from a species that lived first in Africa and then dispersed in Asia, roughly around 2 million years ago, maybe a bit before. And during the early Pleistocene, let me check if I can, yes. During the early Pleistocene, we have a very homogeneous population almost everywhere in Asia, at least from what we can see from the cranial and dental remains. We can find very similar morphology both from the continents and in Indonesia and in Southeast Asia. So as you can see here, the dental morphology is relatively similar. Of course, for those of you who are not very familiar with teeth, they may seem very different, but the main features are there. They are, like, for example, this oblique crest, which starts distally to this cusp. It's also found here. So basically, we have a very similar homogeneous population. But then from the early to middle Pleistocene, there is something happening, like in the continent, in China, notably. You will see that the morphology doesn't change so much. It keeps the main features that were present in early Pleistocene populations. But in Indonesia, you can see some very clear changes, and the morphology is very, very different in Middle Pleistocene dental remains, and also in the few cranial remains that we have. And this is important because you will see later in my talk that there are very specific things happening in Southeast Asia, especially due to island evolution in islands. So let's go back to the late Pleistocene. So these are some of the crania that we know probably represent the latest occurrence of Homo erectus in Asia. So these are from the sites of Ngandong, Sambumachan, and Gawi also. And the site of Ngandong has been recently revised for the chronostratigraphy. And here you see that the site and the layers that yielded these fossils, some of these crania, has been redated to 120 to 110,000 years, more or less. So it's a very late survival of erectus in Indonesia. On the continent, around 300 to 400,000, you find the latest erectus, but after that, there is no evidence of for Homo erectus on the continent. So in island Southeast Asia, you can still see, find some late erectus remains. And keep in mind that this range, and also that some of the specimens have been dated with gamma ray spectrometry. So it's not very clear if these data are secure or not, but if they were correct, then it means that Homo erectus might have survived as late as, let's say, 80 to 60,000 years ago, so which is very, very late. So let's say in the range between 120 and 80, Homo erectus might have survived in Indonesia. And now I would like to go back a bit in time to mention another, what's happening in another island close to Java. It's the island of Flores. So around 1 million years ago, we find some stone tools in a couple of sites. And it's unclear which species did these stone tools. But what is very intriguing is that by 700,000, in the same site where these 1 million years old stone tools were found, in a slightly more recent layer, you can find these kind of remains, which are very small fragments of mandible and very small teeth. And here, for comparison, is a mandible of another species, which I will mention later, which is called Homo floresiensis, and it's known to be a very small-bodied hominin. And you can see that these mandible fragments are even smaller than this mandible. So it's a very very small individual. It's not fully sure if it's Homo floresiensis, but if it's Homo floresiensis, it's much older in time, around 600 years older. And like I will explain to you a bit more the evolution of Homo floresiensis later, but what is very intriguing is that if it's Homo floresiensis, it means that it evolved very early. So now let's go to these endemic species. So as I mentioned before, we have very small body hominin in Flores, and Liangbua is the site where floresiensis has been found. And I will also speak about Calao Cave, where another endemic species from the Philippines has been found recently. So first, Homo floresiensis. So as I mentioned before, Homo floresiensis might derive from kind of hominin that was living already 700,000 years ago on the same island. But the other site, Matamenge, is an open air site. And in this case, the Homo floresiensis remains were found at Liangbua Cave. So you can see it's a pretty large cave. And in the sediments, they found in the upper layers some modern humans. But digging deeper and deeper, they found these remains of several individuals. And they are very small-bodied individuals with a morphology that is not like that of Homo sapiens. So it was a big discovery when they found it. And as you can see, the morphology is clearly different from sapiens. It's much smaller. Here are two mandibles that fit into the ends. And also the chronology is very surprising because it was first supposedly dated to around 20,000, 18,000. But recently, the stratigraphy and dating were revised, and apparently the more secure chronology for these remains would be around 50 to 60,000. Now, by 50 or 60,000, you will see later that there are a lot of other hominin species living around in Southeast Asia or in the continent. So as I said, these Homo floresiensis remains, they show very unusual morphology. The cranium, as you can see, is very rounded, it is very small also. It's quite different from the late erectus that we found, that were found at Gandong, Samumachan, and so on. But they are roughly the same age, maybe a bit younger. And as you can see also, the mandible, even if it's small, it shows a very shallow, very low corpus, and the long bones are relatively small too. So it's a very unusual combination of features. It has been reconstructed in this way with some people suggesting that these small-bodied hominins, they could derive from some australopiths, that would have arrived from Africa at some point, either during the early or middle Pleistocene. The problem is that in Africa, we don't know any australopiths as latest as the middle Pleistocene so far. So it would mean that if they arrived during the early Pleistocene, they stayed in Asia somewhere, and we have no other remains to attest of the presence of the ancestors of these species. An alternative hypothesis is that Homo floresiensis derives from Homo erectus that was living in Java and very close in other islands. And that's the hypothesis that I will favor so far. In the next few slides, I will show you why. And especially because the teeth are very different from those of the Australopithecus and also from Homo habilis. And Homo habilis now is said to be very close to Australopithecus in size and shape for many features, especially for the teeth. And you can see here that the teeth of Homo habilis or those of Australopithecus, which are very similar, they are much more elongated, even if we don't take into account the size. Also the morphology, it's more complicated. We have the hypoconylid that is clearly present. It's not present in Homo floresiensis. So basically, there are a number of features which do not align with Homo habilis or with the australopiths. However, in Java, in the middle Pleistocene sediments, we find these kind of teeth, which are much smaller and more simplified than the classic erectus teeth. And as you can see, they are relatively similar in shape and size to those of Homo floresiensis. So my guess is that it is more likely that a late erectus would be derived into a kind of Homo floresiensis than deriving from Homo habilis. And now I will bring you to the Philippines. And the Philippines are located in a very different situation compared with Flores Island, because it's east of the Wallasey Line. And it means that you have a very different fauna, and for the simple reason that there is no connection, neither now or in the past, between the continents and these islands there. So it means that the fauna, the animals, and the hominins reached the Philippines and these islands here. They necessarily had to cross some water. And this is also a very interesting case, because in northern Luzon, in the north of the Philippines, we have more or less at the same time as in Matamengue in Flores Island, we have evidence of the presence of hominins with some cut marks and stone tools, which are roughly dated around 700,000. So whether it's the same kind of hominin that lived in Flores and then crossed and reached the Philippines, or maybe some kind of Homo erectus that was also present in the same area, we don't know. But what we know is that hominins were there in the Philippines, and it's a very, very important discovery, because so far it was assumed that the Philippines, the first hominins in the Philippines were modern humans around 1,500,000. So you see that there is a pretty big gap now, and hominins reached the Philippines very early. And not very far from Kalinga, where the site with these fauna and cut marks and stone tools, in Kalau Cave, which is located on the cliff of a hill, and on top you have the Kalinga Plains, and then here you have some rivers. And this cave yielded a number of fossils that I will just show you after that, which are very unusual. And I remember I was in Kalau Cave in 2007 doing my master research, and I had the chance to go to the Philippines to excavate the Tabon Cave, another cave I will speak about later. And we visited Kalau Cave, and at that time they just found the first remain of what we know today as Homo luzonensis. And I remember the face of the colleagues. They didn't know what it was. I mean, the morphology of the metatarsal was very unusual for a modern human. They even thought that maybe it could be some kind of carnivore or something else. Eventually, we figured out later that all of these bones belong to hominins, of course, and especially based on the teeth, we could identify it as a different species. So all of this material is relatively small in size. So it belongs to a small-bodied hominin, very similar to Homo floresiensis. But there are also some differences both in the postcrania, but also in the teeth. And notably, the premolars are relatively large in Homo luzonensis compared with the molars, while in Homo floresiensis, the premolars are proportionally smaller. So these differences, plus a few others more subtle, based on the internal morphology of the teeth and also on some morphological features of the postcranium, led us to identify it as a new species that was endemic to the Philippines. It's probably closely related to floresiensis. It shows a number of similarities, but also some differences. So here you have a view of all the dental remains that were found at Kalau Cave. And very strikingly, the dental morphology looks like that, like a modern human for some aspects, like the crown of the premolars and molars. It's very simple. There are no accessory features. It's very modern-like. But the roots, especially of the premolars, are very complex with 3 roots. Normally in modern humans, you have 1 or 2 roots at most. So it's a combination of primitive and more derived feature, and the modern-like features are probably related to allometry, so the reduction in size. So the same process happened independently in Homo sapiens and in Homo luzonensis. The ancestors were with larger teeth, and reducing the size of the teeth, you end up with some similarities. And when the teeth were analyzed, especially the dentine just below the enamel, because it's well preserved and not affected by occlusal wear, we could identify some affinities with erectus, meaning that Homo erectus was the closest species that from which these specimens from Homo floresiensis and Homo luzonensis could be. There are also some sometimes intermediate with modern humans, as I said, probably related to the allometric signal, and they are different from Neanderthals, of course. I didn't include that in this analysis Homo habilis and Australopiths, simply because when I did, they are plotting so far away and so they are so different that it doesn't even make sense to include them in the analysis. And interestingly, both the dentine that I showed before and the pulp chamber also show the same signal. So here, Homo luzonensis plots very closely to Homo erectus, and it's very different from modern humans and Neanderthals. So overall, the dental signal clearly shows that compared with what was expected by some colleagues, that these 2 species could derive from habilis, is very unlikely. And the most likely hypothesis from what I could analyze based on teeth, but also based on other elements, I can mention it later if you want. The Homo erectus hypothesis is the most likely. So Homo luzonensis and Homo floresiensis, they probably derive from some Homo erectus ancestors. Sorry. So now I'm moving from Homo erectus to Denisovans. So very often, Denisovans are qualified as an elusive group. We don't know very well what they are and how to identify them. And of course, the first specimens identified for Denisovans came from Denisova cave in the Altai mountain in Siberia. And there are very few remains that were identified until now. The most diagnostic for the morphology are these two permanent molars. There is also a slight, small piece of distal phalanx and deciduous tooth and a few other remains that have been identified, but the most diagnostic are these two permanent molars. There are also Neanderthal remains that have been identified based on genetics. And this is a very exciting case. You can see it's half red, half blue. It means that it's a hybrid. It's the first generation hybrid from Neanderthal mother and the Denisovan father. And of course, if you were to look at this piece of bone, which is roughly 2 centimeters long and without any morphology, you wouldn't even know if it's a hominin or something else. But thanks to the progress in paleogenetics, now we can extract DNA from this small piece of bone, and we know that it belonged to the daughter of Neanderthal mother and Denisovan father. And it's dated to around 90,000 years old. So it's also quite old. So all of this to say that we know a lot of the Denisovans based on genetics, on molecular analysis, but we don't know much about them based on morphology. Some geneticists proposed to map the morphology using the methylation properties of DNA and saying, okay, maybe based on DNA, we can try to figure out what Denisovan could look like based for the morphology. But all the features that you can see here are actually not very diagnostic. You could say probably that most of these features are the same for many Pleistocene hominins. So it's potentially interesting, but not very useful to identify Denisovans. A few years ago, this piece of mandible was restudied, reanalyzed, and especially combining paleoproteomics and morphology, morphometrics. And the paleoproteomics suggested that it's very closely related to the Denisovans from Denisova cave, so it would be a Denisovan. Now, there are only 2 variants based on paleoproteomics, so these results has been debated. But as you will see, the morphology in this case can be particularly important. So when the paper was published, we were lacking evidence that this could be morphologically a Denisovan, and the result, the interpretation was mostly supported by the paleoproteomics. And so, as I mentioned, we know a lot based on paleogenetics, and we know especially that Denisovans are the group, the sister group of Neanderthals. They are closely related to Neanderthals. So Neanderthals lived in Western Europe and up to the Western Asia. The Denisovans probably lived in most of Asia, and I will come back to that later. And as you see here, there is also some contribution of an unknown early hominin that interbred with early Denisovans. And now, if you think about the geography and time, was the other species living in Asia during the early and middle Pleistocene? Homo erectus. So Homo erectus is the most likely candidate for this ghost lineage. Of course, we cannot demonstrate it so far, but that's something that could be done sometimes in the future if we can get some erectus DNA. And also, we have evidence based on the genetic analysis of modern and living populations of modern humans. And what we know is that Neanderthals contributed to most populations outside of Africa, and Denisovans also interbred with the ancestors of modern humans in Asia and Oceania. And actually, we know that there are up to 3 events of interbreeding between the ancestors of the living populations in Southeast Asia and Denisovans. So now it's not very clear where these interbreeding events happen. Did Denisovans go up to some of these islands in Southeast Asia, and that would be supported by genetics, by the geneticists, because the highest level of Denisovan genome in the modern population is found in the Philippines and in New Guinea. So for them, it suggests that interbreeding happens somewhere in Southeast Asia, and that remained highly concentrated, let's say, in these populations. But actually, the ancestors of modern humans, of the living populations, could have also met the Denisovans somewhere in the continent, and that cannot be excluded, even though the geneticists, they tend to prefer the hypothesis that they met here in Southeast Asia. So now we'll bring you to Laos, and you will see that there are not only the landscape is fantastic, but there are very interesting sites, and especially this site, which is called Cobra Cave, where a few years ago we found this isolated tooth. So when we found it, I didn't know what it was. I said, oh, it's a very weird morphology, definitely not modern human, potentially late erectus, but we didn't know the chronology, we didn't know anything. So we started working on that. And so the tooth come from a very hard breccia that you can see here. Not much of this breccia is preserved, but inside we found a lot of animal teeth, as well as some calcitic floors on top and at the bottom. So we were able to constrain the chronology, and also with direct dating on the teeth of the animals, we know that this tooth now is dated to roughly 160 to 130 years. And now, when we did the analysis, and especially when I was working on the dentin below the enamel, I could immediately tell, okay, it's not erectus, definitely. If you look at Homo erectus here, it's very different in shape. It has this, what we call mid-trigonic crest, which is very often found in Neanderthals. And the shy mandible was just published at the time we found this specimen. So we had a chance to compare with the Tibetan specimen that was attributed to Denisovan based on paleoproteomics. And this second molar in particular shows a lot of common features with the tooth we found in Laos. So now the problem is that this tooth has an extra root below this part of the tooth, and this area here is expanded, which is not the case for the M1, and it's not the case here for our tooth from Laos. So that's why in the geometric morphometric analysis, when we did quantitative analysis of the shape, actually the second molar is falling closer to erectus, while the first molar and the tooth from Laos, they all together, they fall close to Neanderthals. So now, if you remember what I said based on phylogenetics, we know that Denisovans are let's say, the cousins of Neanderthals. And what you can expect based on morphology, especially of the teeth, because the dental morphology and the dentin morphology is very, very important for taxonomy. And you can tell also the phylogenetic relationships based on dentin morphology. So if this one is a Denisovan, then that one also from Laos should be a Denisovan. And you can see that it's quite close to Neanderthals, but a bit outside. And we also had the chance to analyze the tooth we found, the Denisovan tooth we found in Laos, to do some isotopic analysis. And what we found is that compared with Homo sapiens specimen that was found in a nearby cave, I will come back to that later, the cobra cave individual and probably their relatives and all the Denisovans that lived in the area, they probably exploited a more open landscape than Homo sapiens later on when they arrived in the area, were mostly exploiting forest environments. And so this is the position of Cobra Cave here. You see it's roughly 25 meters above the alluvial plain. And here we found a new site, a new cave. And in this cave, again, we found some breccia. And in breccia, it was this year, we found this tooth, which is a hominin tooth. It's not so obvious because it's worn and damaged, but we got some preliminary analysis and it's definitely hominin. So now we are working on it. And we want to thank all the team working in Laos I'm working with, because it's a fantastic research team and the sites are really exceptional. So if you think that the specimens I showed you come from these two sites, and Tampaling, which is a site with modern humans, is on top of the hill, and we still have a lot to explore just on the single hill. And it's full of hills there with this kind of karstic systems, with breccia, with sediments. So there is a huge potential in this area to find much more than what we found. Speaking of Denisovans, I will continue to explain a few things based on this specimen. So Hemianthropus pei, what it was described at the beginning by von Koenigswald in 57, is based on the description of a few teeth. So the von Koenigswald gave a name, a species name. He also did a description, a comparative description with some other hominin teeth and so on. He described the holotype, this tooth, which is called CA673. I will come back to it. And finally, he changed the name very quickly the same year, because he realized that Hemianthropus was already taken, so he called it Hemianthropus. So why am I mentioning Hemianthropus pei, this tooth, this weird tooth? Well, it's because independently from what I'm doing in Laos and other sites, I started working on a collection at the Senckenberg Museum called the Chinese Apothecary Collection. So it's a collection of teeth collected by von Koenigswald in Chinese drugstores in almost everywhere in Asia, mostly in Indonesia, but also a bit in China, Philippines, and so on. And we know that most of these teeth in this collection, they come from southern China, from different caves. We don't know the context. The chronostratigraphy is totally unknown, but we know that they're from southern China. And in 2018, Smith and collaborators analyzed some of these teeth from this collection, and they found out that they have thin enamel, and including this specimen, which is the holotype of Hemianthropus pei. The dentine morphology is unclear between orangutans and humans, so that was not very diagnostic. But what they could say based on analysis of the enamel is that the periodicity is between 9 and 11 days. Now, this is the frequency at which the enamel grows, and in humans, in general, the average is around 7 to 8 days. But in this case, you can see that it's much longer than what we know for most humans. So based on all of this, the authors concluded that enamel thickness, enamel dentine junction, and especially the periodicity of enamel are more similar to Pongo, and thus Hemianthropus pei might be some kind of fossil Pongo. Now, when I started working on this, well, I couldn't find any similarity with Pongo, morphologically speaking. And if you see here extant and fossil Pongo, this is just an example, but if you don't take into account the size, because Pongo can be very small or very large during the Pleistocene, but you can see that the morphology is very different. The dentine horns are much smaller in Pongo. It's more prominent here, and it's more reminiscent of what we see in humans, and especially in Neanderthals. So now these tooth come from southern China, probably middle Pleistocene to late Pleistocene, we don't know. And it's a very similar morphology, it shows a very similar morphology to Neanderthals. So I guess you mean, you know where I'm going now. So I did some geometric morphometric analysis to try to see if it could be Pongo, and it's clearly not Pongo. You can see here that the dentine shape is really, really far from Pongo. Here it's Pongo and here it's Homo. So it's very clear that it's not Pongo. So now if we analyze only humans and we compare only with humans, including erectus, Neanderthals, and modern humans, it's a bit difficult to compare with Denisovans, because for Denisovans we only have M2 second or third molars. It's not very clear. So if we compare with first molar, because this one is very likely a first molar, it's intermediate in shape between Neanderthals and erectus. But if we include it as a second molar, virtually, you can see that it falls not very far from these tooth, which is from Denisova cave, not very far from Neanderthals, in the same morphospace as Denisovans. So in my view, this is very likely a first molar of a Denisovan individual that lived in southern China during the middle to late Pleistocene. And you can see that both in shape and size, it fits with the second molar from Denisova cave. And if it's confirmed, if it's really a Denisovan, then we have a species name for Denisovan, because this is the species that was described in 1957 based on this tooth. And of course, Emanthropus would disappear. It would become Homo, and it would be Homo pei, the holotype of Denisovans, because it would have priority. So moving forward with Denisovans, this is a very interesting case in Sushiayao. So it's a site in northern China, where you can see where they found both upper molars and lower molars, and also some cranial remains. And what you see is that the second molar of Sushiayao is nearly identical to the second molar from Denisova cave. Now the lower molars, especially the dentine, is very similar to the dentine morphology of the Shai mandible molar and to the Cobra cave specimen. So now we can link the morphology of upper and lower molars, and we can say for sure that these belong to the same, probably even to the same individual, but at least to the same group. And we have now information not only for the upper molars, but also for the lower molars, and we can confirm morphologically that these are all likely Denisovans. And again, I mentioned several times in my talk that we can expect for dental morphology, which is very conservative, to find a kind of Neanderthal-like morphology for Denisovans. But it's also the case very likely for the inner ear. The inner ear, like the teeth, they form very early during the growth of the individual, so they preserve well the phylogenetic signal. And you can see here that this inner ear from Sushiayao shows a very high canal, semicircular lateral canal, just like in Neanderthals, while in modern humans, compared with this one, it's much lower. And if you conduct some analysis, you can see that the Sushiayao specimens actually fall at the border of the Neanderthal variation. So again, we have now, morphologically speaking, a lot of ways to identify Denisovans in the Asian record, in the Asian fossil record. So if we do comparative anatomy and we just based on morphology, we compare the different specimens we know are very likely to be Denisovans, like the Sushiayao remains. Then the Shai mandible and the Cobra cave tooth are very likely Denisovans. We have also this this specimen, which is a mandible fragment from Penghu, from the Taiwan Strait. And it shows an accessory root, exactly like the Shai mandible. It doesn't have the third molar, exactly like the Shai mandible. It's not lost, it just didn't grow. So a number of morphological and developmental features, which very likely also suggest that this Penghu specimen is also a Denisovan. And now Arbin. So Arbin, I never saw the tooth, the only one that is preserved here. So I cannot say for sure that it's a Denisovan, but the shape of the cranium and a number of features for me align well with what I would expect for a Denisovan. There are some Neanderthal-like features, I would say. The same for Dali and possibly Maba and Jinushan. So I would definitely assign these to Denisovans because there is comparative evidence based on morphology, and possibly Arbin and some of the crania I mentioned could also be Denisovans. Now there are a few specimens in China which are also middle to late Pleistocene and with very let's say intermediate morphology. These specimens from Wallong Cave, for example, they fall in the middle of the variation of all the Pleistocene and modern humans, and I mean, far from living humans, but in the middle of all of the Pleistocene humans. And the same for the morphology of some part of the cranium of Sushang, which, as you can see here, is in the middle. So it's overlapping with Neanderthals, with modern humans, and a number of things. So not very informative, not very taxonomic, diagnostic in terms of taxonomy, but these could be also Denisovans. I have to say that the dental morphology is very reminiscent to me to what I see in the Cobra Cave tooth and as well as in the Shai mandible. So my guess would be that these are very likely Denisovan individuals. Potentially these also, because we have the inner ear, and as I mentioned, the inner ear can be informative, and here you see that this semicircular canal is quite high. So potentially these are also Denisovans, but they could also belong to some kind of lineage that could be something we don't know, or some hybrids, whatever. And then moving from Denisovans to another species now, the modern humans. So modern humans arrived from Africa. We don't know exactly when, it's unclear. Probably at the transition between the middle and late Pleistocene. And it has been debated for years now that modern humans were in Asia as early as 100 or 120,000 years. But now we have more and more evidence that it's very likely the case. So among some of the candidates for the earliest modern humans in Asia, we have this tooth from Punung, but the chronology is very insecure. It's based on the fauna and a few old datings. Also, the fauna is mixed. At Punung, it's a karstic mix of, well, a lot of things. And you can find also some very recent Holocene teeth inside. So okay, I would not be really relatively sure that this is definitely the earliest modern humans, but it's quite old, and the taphonomy and the color and everything of the tooth suggests that it's quite old. So it should not be discarded yet. The Shiren mandible, while the dating also has been debated a lot, I mean, I'm not an expert in that, and I think the stratigraphy would need to be revised again, but it could potentially be one of the oldest modern humans in southern China and in Asia. These teeth, now that there are like more than 100 teeth from Daoshan, they have been found recently. The chronostratigraphy seems quite robust to me, but the teeth are very strange in a way that they are looking very modern-like. And when I say modern, it's they could be nowadays human. So if you compare the morphology of these teeth with specimens from Africa or Western Asia, you would see that they are very different. They are much more derived and more, modern-like than the specimens from the same age in other regions. So it's possible that they are the earliest modern humans in Asia, but it means that they are already very derived from what we see in Africa, meaning that they might have arrived even earlier. The same for the Luna Cave in China. The dating, has been debated, but it seems to be roughly in the middle of that. And the teeth, not very, very diagnostic. They are supposedly modern humans, but I would like to see more of these teeth, honestly. For these a few teeth, this couple of teeth, actually, the problem is not so much the chronostratigraphy, or at least the chronology, because it was done in a very secure way. The problem could be some reworking or some intrusion of these teeth, but when they worked on the chronostratigraphy, they couldn't figure out any evidence for reworking. So, well, these could be early modern humans. They are definitely modern humans. They could be as old as 70 to 60,000. And finally, one of the oldest one from the Philippines, that this time is from Taban Cave. As I mentioned, I had the chance to excavate there and to be there in 2007. So my colleagues redated some of the human remains, and they found out that, well, this one is probably as old as 50,000. And now we'll bring you back to Laos, where huge work has been done in this cave, Tampaling Cave. So I mentioned it before, but it's on top of the hill where Cobra Cave and the other Tamsot Cave are located. It's a very large cave in which you find a 7-meter deposition of sediment. And now there is a very, very secure chronostratigraphy that has been conducted, a lot of work, using a lot of different methods. And what we know is that the more recent stuff is around 46,000, while the oldest modern human remains that were found were around 80 to 90,000. So these fragments of cranium and maxilla, as well as the mandible, they are among the most recent stuff, but still, you see, they are dated roughly around 50 to 60,000. So it's more or less what one would expect based on genetics, that it corresponds to the out of Africa. But already at that time, the age and the chronology of these specimens were debated. Some thought that it was already too old at 60,000. But now we know that humans probably reached Australia by 65 or even earlier. So it's not so much an issue now. And we also found older evidence for modern humans. And like, for example, this piece of cranium, frontal bone, it's dated to more than 70,000. And there is also a fragment of tibia that is dated to more than 80,000. And interestingly, this frontal bone is quite gracile. And my colleague Sarah Friedlein analyzed the shape of this frontal bone and together with the other frontal from the more recent layer for 50,000 year layer. And what you see is that the 50,000 years layer specimen actually show a more archaic, let's say more primitive-like shape than this one that is older. It's actually more similar to what we see in modern day populations, or to some extent to some of the archaic modern humans. So it doesn't seem to be so easy to identify the evolutionary trends, because the latest modern humans that we find in the same site are more robust and more primitive-like, let's say, than the earliest ones. So it could be related to some local evolution. We know in Southeast Asia that modern humans are very robust during the late Pleistocene. And in Africa, actually, most remains are relatively similar to that one. So we could hypothesize that a group constituted by these kind of individuals came from Africa, and then evolved locally later on. But we cannot exclude that the other specimens, which are more recent, also come from other dispersals and more recent waves of dispersals. So now if I summarize everything, we have at least 6 species of humans that existed in Asia during the late Pleistocene, mostly during the early late Pleistocene. So we have Neanderthals, mostly in Western Asia. Then Denisovans, as I mentioned, many of these sites are Middle Pleistocene, but some of them are also later in time. We have also, of course, the latest evidence for Homo erectus and some probably derived erectus-derived species such as Homo floresiensis and Homo luzonensis, and of course the latest arrival of modern humans in Asia. So it means that there was probably some encounters. We know from the genetics, the paleogenetics and genetics of modern populations that Denisovans, Neanderthals, and modern humans met each other. It's not clear if the latest erectus or these endemic species from Flores and Luzon met the earliest modern humans that arrived in these islands. But what we know for sure is that nowadays only us modern humans survived. And I would like to thank all of the colleagues I'm working with, and without whom this work would not have been possible.